Dr. Helms, we're really excited to have you with us here today.
I'm excited to be here.
We would love to hear about some of the new developments that are coming down the pipeline in your lab.
Oh, I'm so excited to tell you about it because this is work that's being done by Stanford undergrads and medical residents and medical doctors as well. And what we're focusing on is a way to think about reducing scarring in order to improve healing results. Can I give you an example?
Please.
All right, so a lot of the people in the division that I work in plastic and reconstructive surgery, they're very interested in having the best outcomes, which means the least impact of the surgery on the eventual growth of the tissues. So what happens now is that when tissues are knitted back together after surgery, a scar develops at the site of the incision. And sometimes that scar seems inconsequential. But other times, especially if it's in a young child, that scar can actually impede subsequent growth of the face, for example, if it's a repair of a cleft lip and palate. And that growth impedance then makes the child have to have subsequent surgeries to rectify the growth disturbance. We're trying to figure out ways to activate endogenous stem cells through a Wnt-based approach that leads to better tissue regeneration, less scarring, better outcomes in the long run.
Great, that sounds very exciting. So for our audience members who perhaps are not as familiar with basic science, can you explain what a stem cell is and how that process can lead to remodeling scars?
Absolutely. Stem cells have two unique characteristics. The first is they can make exact copies of themselves. That's a process called self-renewal. The second is that a stem cell can become anything. It can become a neuron, a muscle cell, a skin cell, anything. Now, those are embryonic stem cells. In adult tissues, and that is anything after we're born is considered an adult, those stem cells have more limited properties. So let's just talk about the stem cells in skin. They can become more skin, they can become hair, they can become sweat glands. When you have a scar, the tissue is defined as not having hair follicles or sweat glands. And so the idea is by activating endogenous stem cells at the site of an injury, and we happen to use a Wnt protein to do that, you can get those cells that are capable of forming hair follicles, sweat glands, and skin, you can get those cells to repair the defect and therefore not have a scar.
I see. And have you had success with these attempts?
Yes, we have. We're always looking to the next hurdle, but we have had considerable success thus far, and we're funded by the California Institute for Regenerative Medicine, or CIRM, which is funded by California taxpayers who had the initiative to see that funding stem cell research was going to have a future, and I'm incredibly grateful to that organization. And we have had success. We can activate endogenous stem cells. These stem cells accumulate at the site of injury and they give rise to new tissue, and it appears that it's working very well in this regard. So we continue on this path.
Great. And how did you first get interested in studying this idea of stem cells and scars?
Well, if we go back far enough, I was initially interested in developmental biology, that is how an organism grows from a fertilized egg into a human being. And as I studied that process over many, many years, and a number of developmental biologists have said this, we started wondering if it was possible that the same way that tissues repaired after injury was like the way that they formed during embryonic development. And so that led me and many other scientists to begin to look at the process of how do tissues heal. And it did seem like it was very much related to how they formed in the first place. There are obvious differences, and of course those are really, really important to keep track of. But the pathways that were activated in response to injury were similar, and that gave us cues to think about if we understood development and we understood repair, might we understand regeneration? And then becomes a focus on the stem cell.
Right, so you're looking at the natural processes of the body to see how you can fix it when it needs repair.
That's perfectly stated. Can I use that as a tagline? That's it. It's activating the body's own repair mechanisms, trying to improve them, make them a little bit better.
Got it. And today at TEDx, you're going to be talking about a topic called Beauty Reconsidered. Can you tell me how that relates to your experiments that you've been working on in lab?
Well, the first thing is when you hear the word beauty, invariably you think about the face. And those two topics, the face and beauty, are linked nearly inextricably. And I'm not going to give away all the punchlines, but the basic idea is that we have to understand how the concept of beauty and our faces became so important. We have to understand that oftentimes our faces can be indicators of underlying disease, and clinicians use that today to diagnose disease. We also have to remember that beauty isn't defined by our faces alone. Beauty is compassion and kindness and warmth. And that's internal beauty. That's the most important beauty. So that's why it's called Beauty Reconsidered.
I love how you bring all these different elements into medicine, which others might think can be kind of stuffy and technical, but you bring all these other elements into it that make it very much alive.
Well, thank you very much. I appreciate that comment.
Well, thank you so much for your time with us and for answering our questions.
My pleasure. A delight to be here.